Vaccinology, which is the science of the development of vaccines, has historically included basic science, immunogens, and clinical evaluation. But more recently, the science has expanded further to include the safety, ethical, and economic considerations of vaccine development. Over the past 100 years, the development of working vaccines for major infectious diseases, such as pneumonia, the flu, and tuberculosis, has contributed enormously in public health. On the right graphic, we can see that within a span of 100 years, basically, cardiovascular diseases and cancers have surpassed common infectious diseases as the leading cause of medically related causes of death. And so computer vaccinology, computational vaccinology, or vaccine informatics is a new and interdisciplinary branch that combines computer science, mathematics, statistics, and immunology in order to answer questions in vaccinology with computer-driven algorithms. And to understand how vaccines work, it helps to look at how the body fights infections. With germs such as bacteria or viruses that invade the body, they multiply and attack. This invasion is called an infection, and it causes illness. Luckily, the body has several tools against this infection. Blood contains red blood cells for carrying oxygen to tissues and organs, and white immune cells for targeting infections. These white cells consist primarily of three types of cells. First, macrophages attack germs and dying cells and leave behind parts of pathogens called antigens. The body identifies these antigens as dangerous and creates antibodies to attack them. B lymphocytes, also known as B cells, are defensive white blood cells that produce antibodies that attack antigens left by macrophages. And lastly, T cells are defensive white blood cells that attack already infected cells. After an infection, the body remembers what it has learned about how to counter the disease with memory cells, which quickly go into action if the same disease comes again, as shown in the second graphic, where on the second time the body recognizes the same pathogen or antibodies, antibodies introduced in a body, the number of antibodies produced are greatly increased. And vaccines initiate an infection that doesn't cause an illness, but does cause the immune system to produce T cells and antibodies. After getting a vaccine, the imitation infection can cause minor symptoms that are normal and should be expected as the body builds immunity. After the body recovers from the imitation infection, the body is left with a large amount of memory T cells and B cells that remember how to fight the same disease in the future. And on the right, those are the 4 basic steps for how the body builds immunity with the introduction of a vaccine. So epitopes. Epitopes are the part of an antigen molecule to which an antibody attaches themselves. They are recognized by the immune system as a peptide or protein and are either conformational or linear, depending on their spatial orientation. Essentially, knowing where these epitopes are on pathogenic cells give us the blueprints of basically its vaccines. And identifying specific epitopes have been vital for the development of diagnostics tests for various diseases and the development and design of vaccines. But there's a large number of computational algorithms and software programs that have been developed for this specific type of prediction. These remain inefficient due to the limitations of today's technology. This type of intermolecular interaction between antibodies and epitopes are used today in the efforts for developing a working vaccine against the coronavirus. With the 2019 coronavirus being discovered so recently, there's really little knowledge regarding its immunogenic epitopes. But it's been found that there's been many similarities to the severe acute respiratory syndrome coronavirus that peaked earlier in 2012 to 2013, as shown on the right. Some examples are its attachment receptors and gene receptors. And on the left, it shows the basic structure of all coronaviruses. And using this knowledge, some studies have combined immunoinformatics with a comparative genomic approach in order to determine a set of epitopes that can be introduced as potential targets for developing peptide vaccines against the virus. And today, there are supercomputers that provide new techniques to combat the virus. In Japan, the Fugaku supercomputer, which is the world's fastest supercomputer, as shown on the bottom left, is capable of performing 415 quadrillion computations per second. And currently, it runs intermolecular simulations to find drugs that can bond with proteins related to infections and the growth and spread of the virus in order to deactivate them. And within 10 days, it has already isolated 12 out of 2,100 existing drugs, and currently all of which are clinical testing. Summit is the world's second fastest supercomputer on the top left, and it is currently performing data mining research that has recently uncovered a common pattern of gene activity found in the lungs of infected patients, the overexpression of a gene that produces bradykinin, which is a compound that regulates blood pressure. And the symptoms of having a hyperabundance amount of bradykinin in various organs and blood vessels mirrors that of the manifested symptoms of COVID-19. And both supercomputers rely on machine learning, which is the study of how computer algorithms can improve with time and experience to become better at simulating molecular interactions, whether it be among proteins or immune response cells, antibodies, and epitopes. And this basically allows scientists to discover important connections that link symptoms of a virus to the genes that affect it. And studying specific molecules and interactions are not the primary purpose of computers in computational vaccinology. So in fact, recent advances in this field has allowed for the creation of an artificial immune system known as the AIS. Purpose is not only to reduce the need for clinical testing in animals, but it's also used to better understand the immune system using computational and mathematical models. It runs on four main primary techniques: the clonal selection algorithm, negative selection algorithm, immune network algorithm, and dendritic cell algorithms, which are all inspired by recent theories in immunology that explain the structure and behavior of all mammals, of the immune system in all mammals. And these techniques combine intricate and specific logic gates of biological conditions. Interestingly, just as the immune system adapts better to new diseases over time, the artificial intelligence behind AIS learns from each introduction of a pathogen or vaccine into its organs. And over the years, with the help of computers and machine learning, vaccinology has transitioned away from solely studying the control of infectious diseases, but rather began making significant advances in the control of infectious diseases such as cancer, non-infectious diseases such as cancers, addictions, and neurodegenerative diseases. Oh boy, sorry. There are currently several challenges in vaccinology that require machine learning to address. First, a three-dimensional 3D prediction of a protein structure crucial for vaccine development is very difficult and lengthy to obtain due to the sheer numbers of possibilities that amino acids can be arranged within epitopes. And furthermore, a successful cancer vaccine requires the identification of antigens that are unique to cancers and their interactions with immune response cells, and that's not possible without the completion of the artificial immune system. Both the Fugaku and Summit supercomputers are built upon previous works on resources from the AIS for data analyses. But besides being a continual learning model, the AIS is a viable contender for being an example of an effective unsupervised machine learning. If incorporated into mainstream technology, the AIS has the potential to greatly advance computational vaccinology and drastically reduce the amount of time to discover a working vaccine. And currently our knowledge of gene markers that predict protective future immunity is greatly limited, hence why cancers have not been cured yet. But we have yet to make drugs that effectively target specific disease-inducing sites. As pathogens become more and more resistant to develop immunities to drugs, it is increasingly important to outpace their rapid mutations and adaptations. Today, it's nearly impossible to isolate on all intermolecular interactions that underlie the causes of symptoms of diseases, but it's clear that machine learning will be a tremendously powerful tool in not only progressing computational vaccinology, but also make significant contributions in public health. Thank you, everyone.